digital display. So drinking water should not have turbidity more than 10 NTU, nephelometric turbidity unit. So that part also was mentioned in the last class. These tests are very useful in determining the detention time in settling for the raw water and to dose of coagulants required to remove turbidity. So, and how turbidity occurs in the soil, that part also has been discussed in the previous class. So, with this, we'll go and we'll try to see what are the methodologies of determination of turbidity in details. So, the first one is obviously turbidity rod, as I mentioned, and this turbidity rod can easily measure the in the field with the it can measure the turbidity in the field, and it can measure very easily. Now it consists of an aluminum rod which is graduated as to give the turbidity directly in silica units per milligram per liter, right? And to the upper end of this rod is attached to a graduated non-stretchable tape. Fine, and it is mentioned in the right hand side photo. You can see that graduated tape, non-stretchable tape, so as to help in lowering the rod. So that rod has to be lowered down. A screw containing platinum needle and a nickel ring is inserted at the lower end of the rod, which also can be seen over here. We have that aluminum rod graduated to directly read the turbidity meter, and at the bottom we have nickel ring and platinum needle also. At the in the right hand side you can see in the figure. Now the standard platinum needle is 1 mm in diameter and 25 mm in length. A vertical rod or a stick is inserted in the nickel ring so as to support and keep the rod in a vertical position, which is very much important to remember. Now, in order to measure the turbidity, the graduated aluminum rod is lowered in the water, and the depth at which the platinum needle ceases to be seen. Now, in the eye mark, we have to keep our eyes. So, keeping the eye at the eye mark is also important. Under standard light condition is noted, which directly gives the turbidity in ppm or silica standard. Fine. So this is the way we can measure, and the length are also mentioned over here. You can see in the figure, and the same has been mentioned over here. You can see that. So this is the overall process or principle by which we can get the turbidity of water by the help of turbidity rod or turbidity tape. Now the next is Jackson's turbidity meter. So this is also another one important conventional instrument through which we can yield the overall turbidity of water. So basically Jackson turbidity meter is also called as Jackson's candle turbidity meter because it works with the help of a standard candle, and it consists of a calibrated glass tube which can be placed on a metallic cylindrical tube holder fixed over a metallic stand provided with the fixed standard candle at the bottom, which can be seen in this particular schematic diagram. So we have that metallic stand, we have that metallic container tube on the top of the stand, and then at the top of that we have a graduated glass tube, and just beneath the stand we have that standard candle holder, and with that actually holds the standard candle vertically. Fine. So what is done is, the water sample is poured in the water sample is poured in the sample holder, and the image of the flame of the standard candle is seen through the turbid water in the glass tube. So this is the usual process. Now the level of water in the glass tube is gradually increased till the image of the flame ceases to be seen. Right? So the height of the water column measured in the graduated glass tube provides the measure of the turbidity of water. Though these are not direct method, but these are conventional one, and they have been used for several decades. And the longer the light path of 21 centimeter corresponds to 100 JTU, so that is Jackson turbidity unit, while light path of 10.8 centimeter corresponds to 200 JTU, where 1 JTU is equal to 1 ppm. Fine. So you can gauge that, if you are able to see longer light path, that means the turbidity is lesser. So that part is also important to remember over here. So we have Jackson turbidity unit also, like what we see nephelometric turbidity unit. So Jackson JTU is actually its equivalent to 1 ppm. Fine. So this is the overall process of Jackson turbidity meter. And the third one, as I mentioned before, so Bailey's turbidimeter, which we are going to talk about next. So in the right hand side, you can see that this is the Bailey's turbidimeter, and we have sections which have been shown, and if you see, it consists of a closed galvanized iron box, fine, and on one side of which two glass tubes can be held vertically side by side. Fine. So left hand side, if you see the section AA, you can see both the vertical rods which have been attached to the bottom blue cobalt glass plate, and the two glass rods are actually attached to that, and which are actually kept vertical side by side. On the other side, in front of the tubes, a 250 watts electric bulb is located with a reflector. So you can see in the right hand side we have that reflector, and we have that 250 watt electric bulb. So what is done is, the glass tubes are supported at bottom by white opal glass plate and are surrounded by blue cobalt plates, so which I have already mentioned. Now one of the two glass tubes is filled with water, and the water whose turbidity has to be measured, and the other is filled with the standard water solution of known turbidity. So we can say that is a standard solution, and the electric bulb is lighted subsequently, and the blue color is in both the tubes is observed. Fine. Now from the top of the instrument. Now if the color of both the tubes differ, the standard solution tube is replaced by another standard tube of different turbidity. Now the process is continued till a matching is obtained in the colors of both the tubes. Now the turbidity of standard solution will then correspond to the turbidity of the sample of the water, and by doing that we can actually tell that this is the same turbidity of the other tube for the samples which we are actually testing. Now Bailey's turbidity meter can precisely record low turbidities as well, so of the order of 0 to 2 ppm. So since the these things actually I have discussed, so this method is we can go to the very precise low turbidity measurement also, since what we are doing is we are basically measuring based on the comparison. So we are basically, you keep on changing the standard solution with known turbidity, and then after a certain point of time you will see that the color is same in both the container, and then you can tell that the liquid which has unknown turbidity will be same as the known standard solution. So this is the overall philosophy, and as we use this, so we can go to the better extent. The minimum range is very well recorded and very precisely recorded in this particular instrument. So apart from this, we have some commercial turbidimeters which I was talking about. So nephelometers we can call them, and so these are actually used in large scale, particularly in measuring very low turbidities of drinking water and less than 1 unit. So what is done is, if you see, like the basic difference between the old and the new turbidimeters is that in the old instrument the light was seen or measured after the same passed straight through the turbid water. While in a modern turbidimeter, the light intensity is measured at right angles to the incident light. Now, such an instrument therefore uses a photometer to measure the intensity of light passing through the turbid water after the same is scattered at right angles to the incident light. Now, the main advantage, if you see, so the process is actually given over here. We have a lamp and then we have a lens, and then the light gets scattered. We have the sample cell, and after that we'll have this transmitted light attenuator, and through which it will be detected in the detector. So, and we have that 90-degree detector also. So with the help of that, so that is basically photometer, with the help of that we'll be able to get, and since we are taking help from different electronic components, we can get very low turbidity with high precisions, right? So since no direct relationship between the intensity of light scattered at 90-degree and Jackson candle turbidity exists, nephelometric turbidity unit has been abbreviated. So that is why nephelometric turbidity unit, and mostly it is used since these days. Mostly commercial turbidimeters or nephelometers are used in most of the laboratories. Also, you notice that these are tabletop commercial turbidimeters, which is also known as nephelometers. Now, commercial ratio turbidimeter can measure the ratio of the 90-degree light scattered to the transmitted light. So at low to moderate turbidity, the forward scattered signal is negligible compared to the transmitted signal, and the output is simply a ratio of the 90-degree scattered light to the transmitted light. Fine. So these are the important thing which needs to be kept in the mind while talking about modern days turbidimeters. And there might be questions. People may ask you what are the difference between modern day nephelometers and all those conventional methods which we have discussed. So you should be able to tell the difference. In any commercial ratio turbidimeter, what are the difference with the one which we have discussed before in the conventional one. So mainly what happens is, in this case we don't talk about the direct light, we talk about the transmitted signals, and the output is simply a ratio of the 90-degree scattered light to the transmitted light. So that part is very important to remember. So with this, we'll move to the next part. So that is the color. So color is also important. Most of you would agree with me, because this gives aesthetic aspect, and that is why color is important. In previous class also we are discussing about colors and how this color can change the perception regardless of whether that water is good, or whether that water is suitable for drinking or not. But if the color is not good, so our brain would not allow us to drink that water. So that is why color becomes very important when we talk about the characteristics, physical characteristics especially of the water. So dissolved organic matter from decaying vegetations or inorganic materials, such as colored soils, may impart color in the with colloidal conditions from decaying vegetables. So these are the several factors which can create some different colors. Sometimes excessive growth of the algae and aquatic microorganisms also cause color changes in the water. So the presence of color in water is not objectionable from health point of view, which actually I was trying to mention, but may spoil the color of the clothes being washed in such waters. So you will see that if you wash a very if you wash colorful clothes, white clothes or cream color clothes, so the color will not be bright after washing, because all this strain will be attached to the garments which you are washing in such kind of waters. And that is why this is objectionable from that point of view. However, what I mentioned, that is also important here, that it is also objectionable from aesthetic and psychological point of view, as people may not like to drink colored waters. So that is why the color in water can easily be detected by the naked eye, no doubt about it, and it can be measured by comparing the color of water sample with other standard glass tubes, so which is known as Nessler tubes. Fine, so containing solutions of that which produced by 1 milligram of platinum cobalt dissolved in 1 liter of distilled water. So that is the way the color can be measured. So the standard unit of color is the basically which is produced by 1 milligram of platinum cobalt dissolved in 1 liter of distilled water. So that is the way we basically define the unit to measure the color's intensity into the water. Now, different standard color intensities representing as 1 ppm, 2 ppm, 3 ppm can thus be easily prepared, by dissolving 1 milligram, 2 milligram, 3 milligram respectively of platinum cobalt in 1 liter of water, and the maximum permissible color for domestic supplies is 20 ppm. Fine. 20 ppm is the maximum color. Now, if we want to get, apart from that, which I have told, that Nessler tubes, if you want to determine precisely small color intensities within a compact instrument properly lighted from inside, then we can take help from tintometer. This tintometer is usually generally used to determine the color. So the instrument contains an eyepiece with two holes, and a slide of standard colored water is seen through one hole, and the slide of water to be tested is seen from the other hole, and the standard color slide is replaced by another till a matching is obtained, and is done in a turbidimeter. Fine, so which we discussed before. Now the standard color intensity corresponding to this matching will represent the intensity of the color of the water sample for which turbidity was unknown. So the maximum ppm is mentioned, and the preferable one is, though the maximum limit is 20 ppm, the preferable one is always less than 10. Fine. So this is the way we actually take care of the color aspect of drinking water, or while talking about water supply. This is the way we maintain the permissible color intensity into the fresh water. Next, another one very important aspect is taste and odor. So if you see, this taste and odor are very important because they have direct relation with how much people will consume, and they are objectionable also if the taste and odors are not normal. So again, if you see, the dissolved organic materials or the inorganic salts or the dissolved gases may impart tastes and odors to the water, which generally occur together. If there is a change in the taste, at the same time you will see there is a difference in the odors, or there will be some foul smell, there will be some sort of stinking in the water. Now, all these often happens because of the dissolved gases such as hydrogen sulfide, methane, and carbon dioxide. So these are the most predominant one which can create a different odor in the water. And at the same time, combination with the organic matter, sometimes mineral substances like NaCl, iron compounds, carbonates, and sulfates of other elements, and phenols and other tarry or oily matter, especially after the chlorination, we may have some this kind of tarry oily substances, and those also contribute to the change in the taste and as well as odor. Fine. So the taste of these are done by sense of smell, and the taste, because these are present in such small proportion that it is difficult to detect them by chemical analysis. The water having bad smell and odor is objectionable and should not be supplied to the public supply. Now the intensity of the orders are measured in terms of threshold number or threshold order number, whatever you say. So these are measured in terms of threshold number or threshold order number, right? Which represents the dilution ratio at which the order is hardly detectable. So this is also a very good philosophy to detect. Like if you get some order in water and if you keep on diluting that water, after a certain point of time, there will not be any odor present which can be detected by our nose. So that is why this is the way the order is actually determined. The number of times the sample is diluted represents the threshold order number. Now, if 40 ml of sample is diluted and is made 200 ml, till it just loses its taste or odor, then the threshold number will be 5. So now you should be able to understand how this taste and odor are measured in case of fresh water. For public uses, the threshold number should be within 1, and it should be around 1, and it should not exceed 3. So that is also important to remember. Next, another one important part is temperature. Now, temperature of water is measured by means of ordinary thermometers. As you can understand that this is the most cheapest way you can measure the temperature of the water, and it would gives sufficiently reliable results. The temperature of surface water is generally at atmospheric temperature, and that is very obvious, while that of the groundwater may be more or less than the atmospheric temperature, depending upon the temperature of the surrounding soils or surrounding geomaterials. The most desirable temperature for public supply is between 4.4 degrees centigrade to 10 degrees centigrade. Now, the temperature above 35 degrees centigrade are unfit for public use, and this water is not palatable, so people will not be comfortable consuming water which has temperature more than 35 degree sorry, more than 25 degree centigrade. Fine. So now we'll be talking about the specific conductivity of water. So the total amount of dissolved salts present in water can be easily estimated by measuring the specific conductivity of water. The specific conductivity of water is determined by means of potable ionic water tester, and as expressed in micromoles per centimeter. Fine. So MHO is the unit of conductivity, and equals 1 ampere per 1 volt. So this is the way it is expressed in micromoles. So and the temperature is also standard is 25 degrees centigrade. The specific conductivity of water in micromoles per centimeter at 25 degree is multiplied by a coefficient which is generally 0.65, so as to directly obtain the dissolved salt content in milligrams per liter or ppm. The exact value of this coefficient depends upon the type of salts present in the water. So this is very these are the points which you need to remember. People would ask you questions. The exact value of coefficient depends upon the type of salts present in water. So these are the important part. You may get multiple choice question, as well as during your placements also you can get questions from this part, because these are tricky and difficult to remember. The small points, but they are very important. Fine. So this is the way specific conductivity of water is measured and helps in understanding the overall physical characteristics of water. So with this, now we are slowly moving into the chemical characteristics of water. The chemical analysis of water is carried out in order to determine the chemical characteristics. This involves test for determining the total solid, suspended solid, pH values, hardness, chloride content, nitrogen content, iron, manganese, and other metal contents, dissolved gases, and there are very standard trace protocols are available, which we will be discussing in this particular subject. And so I have enlisted the several parameters which we are going to talk about subsequently in today's class. So let us talk about first total solid and suspended solid. So these two are very important as we talk about chemical characteristics of water. If you see, total solids includes the solids in suspension, colloidal, and in dissolved form, and the quantity of suspended solids is determined by filtering the sample of water through fine filters, drying, and weighing the samples. So the quantity of dissolved and colloidal solids is determined by evaporating the filtered water obtained from the suspended solid test and weighing the residue. So you will get all three parameters from all two parameters, total solids and suspended solids, as well as colloidal solids also you will get. And the total solids in a water sample can be directly determined by evaporating the filtered water obtained from suspended solid test and weighing the residue. The total solids in a water sample can be sometimes by we can what we can do is we can put that entire water into the muffle furnace, and then after getting it fused in the muffle furnace, we can check the weight of the residue in order to get the total solids present into it, because whatever organic solids will be there will get decomposed in the muffle furnace, and whereas only inorganic solids will remain. Right? So by weighing, we can then determine the inorganic solids, and deducting it from the total solids, we can calculate the organic solids. So there are several ways by which we can get the values of total solid and suspended solids. So as soon as we know the total solids present into it, we try to get the knowledge, or we try to get to measure the inorganic part. Now, once inorganic part is known from the muffle furnace test, we can directly get the organic part by subtracting it from the total solid. So, I guess all of you have understood how this method is done to calculate the total solid and suspended solids. Now, this characterization and most of them you will be practiced in the laboratory classes or practical classes, maybe this time by virtual mode, but still you will have a better idea when you see those animations and in the virtual lab activities how to determine total solids, suspended solids, and other tests which we usually perform in undergraduate environmental engineering lab, determination of pH, determination of DOD, and so on. So we'll be discussing them, and the same thing, that time what you can do is you can go through these lectures again, and then you can go to that particular lab. It will give you better connection and as well as better understanding. Now, second very important part is pH. pH is that indicates whether the water is acidic or alkaline. So pH is basically, it's a logarithmic of reciprocal of the hydrogen ion concentration present in water, and that is the reason it talks about whether the water is acidic or the alkaline. Now, lower value of pH means higher hydrogen ion concentrations representing acidic solutions, and if you see the bottom part, we have actually given a scale. So usually 7.5 to 8.5 is considered as neutral, and the more you go, the more pH you get, the alkalinity would increase. In the contrary, once the pH is lesser, the acidity of that particular solution would increase. So this is the usual way by which we can. So 6.5 to 8.5, this is the range where we say that this is neutral, and when it scatters from this particular zone, either it has to be alkaline or it has to be acidic. Now, pH value of water is generally determined, so depending upon the nature of the dissolved salts and minerals, the pH value range from 0 to 4, and we have discussed all those things. The lower value may cause tuberculation and corrosions. Okay, fine. High value may produce incrustation, sediment deposits. That is also true. Now, pH value of water is generally determined by pH papers or using pH meter. So these days we get pocket pH meter, tabletop pH meter, which are very, very sophisticated and can give very correct results. Now, pH can be read directly on scale or by digital display using a pH meter. So that is what actually I was talking about. So we may have several problems associated with pH, and we'll solve those problems later on. Now, we'll be talking about measurement of pH. So we have basically two different methods by which pH can be measured. So pH is, it is very important. The moment we get a liquid, we first try to determine the pH to understand whether this is alkaline or acidic. So its knowledge is also helpful in controlling the softening and coagulation process in water treatment. And there are two methods of determining pH value of water. These are not method, mostly we should say the principle, and then based on these principles we have several different devices by which we can measure the pH. The first one is colorimetric method. So this is probably the most simple method, in which some indicator is added to the water sample and the color of the solution of the known pH value. So what happens is, these standard colors may be in the form of colored liquids in glass tubes, or colored glass discs, or colored charts supplied by the manufacturers with each indicator. Now, for acidic range, the common indicators are thymol blue, bromophenol blue, methyl red, and chlorophenol red. So these are the within the acidity range, we can use them as common indicators. Whereas for alkaline range, the common indicators are thymol blue, phenolphthalein red, and toluene red. So in alkaline range, we can use these particular chemicals to indicate the pH value. So this is the way we can basically determine the pH with the help of colorimetric method. So the moment you use them, there will be change in color, and based on the color charts provided by the manufacturers, we will be able to get the value of pH. Fine. Now the second method is electrometric method. So in this method, pH meter is used, as I was talking about. Mostly in nature, they are potentiometer. So the specimen of water is kept in a beaker, and the two electrodes of that instrument are dipped in the water, connected to a dry cell, and the current passing through the circuit indicates the pH value, which is read directly on the dial. And the electrodes commonly used are made of calomel glass or quinhydron. These are the different material by which the electrodes are made. And in some of the latest pH meters, or modern days pH meter, the pH value is displayed electronically on the dial up to the second decimal places of the value. So that's what actually I was talking about. In modern days, we don't do other tests, simply we have a probe. So within that, everything is done. So you dip that probe and you wait for certain time, and then directly you get the results. So whatever is happening within and at the same time, through that probe we have that sophisticated microprocessors which is attached, and directly we get the value of that. So that probe is able to detect the hydrogen ion concentration present in that particular liquid. So with this, I'll stop today, and in the next class we'll continue with the hardness and the determination of hardness and influence of hardness in the chemical characteristics of water.